Hello guys, my name is Dr. Varun and we are back with 30 days, 30 most important topic for NEET 2025 biology.
Chalo, let's complete it.
In today's video, we're gonna be talking about the application of biotechnology. It is a high weightage chapter. In fact, this whole unit is very high weightage because 9 to 10 questions will definitely come from this unit. So two topics which I want to make a more emphasis on this chapter is the tissue culture and the molecular diagnosis. Molecular diagnosis, 100% questions coming chances are there, whereas tissue culture could be the second most important topic. So let's just start with the tissue culture. Now, what is tissue culture? Basically, what happens, we take the certain cells of the plant and we try to grow them. How? We know the hormones of the plants. So there are certain hormones which will grow the shoot, certain hormones which will grow the roots. Which do that? Your root will be grown by the auxins. Cytokinins will grow what? Your shoot part of it. Okay, so this will help us to grow that part. We will supply the nutrients and everything and grow the cells. So this is what our basic understanding that what happened inside the tissue culture. So in tissue culture, what we are doing, we are taking certain cells of the plant. So imagine that this is your plant which you want to grow. So what we will do, we will take certain cells of it. Certain cells, when we are taking it, those group of cells is called as what? It is called as callus. Now this callus is put into the artificial medium. Here I am making a petri dish. In that medium I have given whatever the nutritive medium is needed. Nutritive medium means your water, nutrients, whichever is required for the plant growth, plus the hormones. So what will happen? These cells will make what? It make a small plant. These small plant later on put it onto the soil and they grow into what? Same like this plant characteristics, because we have taken the cell of this. Now these smaller plants are called as what? We call them explants. And the ones which now when they will grow up, they will look like what? They will look exactly like their parents. So they are what? Soma clones. Let's just see that what NCERT furthermore saying about it. NCERT told about this way of growing the multiple plants in shorter period of a time is known as what? Known as tissue culture. Okay, and this whole thing, since we are using the smaller part of the plant and growing it, called as micro propagation method. Okay, moving to the next thing would be that how this the cells which we have taken has the capacity of forming any part of the cells, because the cells which we have taken, they have totipotency, means they have total potential to make what? In all the parts of the stem, roots, leaves of the plant. So this is one such thing. Moving on further about the tissue culture, the next thing is that sometimes what happened, we used to like if someone will ask like which plant we have made potato, tomato, sugar cane from this. Okay, now if we coming back that if we want to make a plant which is free from disease, then we will take what? We will take the meristem of the plant. So meristem could be the shoot apical meristem or the root apical meristem. So the next cultivation is merismatic or meristem culture. So in this, what we are doing, we are taking the meristem to grow the act like a callus, and then we will grow the explant out of it. Why we are doing it? Major question is why. The answer to the why is that meristem is free from viral infections. So means they grow so faster that the virus is still not has been able to infect those cells. So we can make what? Pathogen free or non-contaminated plant. Okay, how this will be helpful? Imagine that, for example, this I am making you understand that, for example, a disease outbreak happened in a region and the plants which are present over there, we have made it with the hybridization. So means they are very important plants for us. We don't want their species to go eroded because of the infection, pathogen infection. So what we did, we take the meristem of those. Meristem is free from the any viral infection. We will grow them and cultivate them, and whole of the crop which has been destroyed, still we are able to propagate it. Understood? This is meristem. The last one is the protoplasmic fusion. So last is the protoplasmic fusion. So basically what we do, we take the cells of two different plants with a desirable characters. Like for example, we take potato and tomato cell. We break their cell wall and fuses their protoplasm. So it becomes what? Single. Later on we fuses the nucleus also of it. After the fusion is done, it forms what? One cell, and then we grow that on the nutritive medium. So what happened? This cell has a feature of what? Potato also, tomato also. Okay, so the new plant that will be forming what? Having both the characteristic. So tomato was formed, but since it do not have all the desirable character which scientists were looking forward, so we stopped this experiment. But we have to remember that we can do this thing also, where we can fuse the protoplasm of two different cells and get the desired characters. So basically this is called as what? Somatic hybridization. Means we are taking the somatic cells and we are hybridizing it, two different somatic cells joined together, and this process is called, is what, like in this somatic, this is a process, and the things that, the new thing that has been formed, that is tomato, that is what? Somatic hybrid. This is all about the tissue culture. So in tissue culture, if we closely look, we said that certain statements, that tissue culture is what? Totipotency, that certain cells have the potentials to form anything. Then explant is what? These are the cells which we have taken from the plant and grown in the nutritive medium, and when they form the small plantlets, germinate, so we call it explant. This whole process is called as what? Micropropagation, and all of these plants which explant that is formed, it will be similar to the parent from where the cell has been taken. We call it soma clones. Then merismatic culture is there, that the meristem is free from any viral contamination, so we use it to make the virus free plants. Then protoplasmic fusion is what? Two different cells of two different plants has been taken and fused together and form a new somatic hybrid. The process called as somatic hybridization. This is about the tissue culture. Next is the molecular diagnosis, which is also one of the important topic. So next is molecular diagnosis. In molecular diagnosis, what happens? Since the conventional way of urine analysis, blood analysis will take lot of time. We don't have that much of time, or it will be done when the full grown thing has been done. So instead of that, what we do, when any disease is there and it has been just taken into the account, then we will do it. Like for example, if it is the case of cancer, for example, sometimes what happen, that you had certain nodule in your body, like female has a nodule in the breast area, so we touch it, and if we feel that there is some kind of lump or a hard swelling present inside the breast of anyone, we take that part and we make a cut section and we check it. When we are checking it for the infection or these things, we are able to find it out whether it's happening or not. Or maybe for the HIV, for example, if person is there, he got infected with the HIV, so we can take that sample, okay, of his blood or serum, whatever, and then we will put it into the process, and the process will tell us, okay, it has the viral DNA of HIV virus. So we are able to find out and give the accordingly the treatments. Okay, prophylaxis can be also given. So moving on, that molecular diagnosis, three different ways we can use it. That is, first is recombinant DNA technology. We have PCR, that is polymerase chain reaction, and ELISA, that is enzyme linked immunosorbent assay. Now you have to remember that this enzyme linked immunosorbent assay, it is used for what? It is used for finding lot of diseases, because it uses what? Antigen antibody reaction. So already there are certain antibodies are present, so antigen go attached to it. okay, or the antibodies are present, antigen will go attached to it. So antibody antigen reactions will occur over it. Okay, so it is used to detect the whether a particular type of glycoprotein present inside the blood or not. Okay, infection. PCR is used what? If the minute quantity of DNA for anything is present, we can amplify it and tell that yes, it is present into it. So polymerase chain reaction, everyone know that we use for the HIV, cancer, all of these things. And recombinant DNA technology is what? That we are using a probe kind of a method to find it out. So probe is what? In this, a radioactive substance has been added into the DNA, and this is what we have kept it. Now, if there will be a counterpart present, okay, so normally what happens? Single stranded probe is being made. If its counterpart will be present, it will go and attach to it, and we are able to find it out. If not, then we are not able to find it out. So this is the method of molecular diagnosis. I hope these things are clear to you. Statements can directly come. Terms like autoradiography and all of these things can come to you. I try to concise it. It is an easier thing to do, but don't left it. That's why I'm taking this. Well, that's all for this video. If you like this video, give it a like, share it with your friends. And if you haven't subscribed, give me a subscribe. That motivates me a lot. Thank you so much. We will meet again.